I wish you very welcome to today's webinar, Combating COVID-19. And my name is Siv Anderson. I'm co-director of SciLifeLab, and I will moderate today's webinar. Today you will hear some of the very important work that has been done at SciLifeLab with the aid of funding from Knut and Alice Wallenberg Foundation and from SciLifeLab. And we are very pleased that Jenny Nordborg, the head of Office for Life Sciences at the Government Office in Sweden, is here to say a few introductory words. Please, Jenny.
Thank you so much for the invitation. It's a true privilege to be able to participate in this conference today and to be able to address you here at the beginning of the meeting. So I just want to talk from a few slides that I have. And to start off, and as we all know, the COVID-19 pandemic really put a focus on life sciences as a whole. It became clear to every individual and the society as a whole how important this sector is for society. It became so evident how the research infrastructures that we normally don't have so much insight in on a personal level, each and every one of us, are really backbones to our society. And I'm very proud of being a small part of that, being part of the life sciences community. It's really imperative that we do join forces and fully utilize the national infrastructures that we have, both in handling the acute situation, of course, and to be speedy in building up new knowledge when it comes to global sharing. The government agencies, of course, were able to man up in a very quick and swiftly manner ranging from the Science Council and Vinnova, from Forte and Formas. We saw a lot of immediate action. But I must stress that the way that SciLifeLab and Genomic Medicine Sweden immediately took action in working with on the daily basis, that was super important for Sweden. The National Strategy for Life Sciences was launched almost a year ago. It feels like ancient times, but it's still so much value also now when we have so many new learnings, because we're building the strategy on a very long-term and a holistic perspective. When we talk about the economic prosperity really leading on the health and sustainability of our society. The priorities that we have set in the strategy are still valid through the pandemic. We have 30 priorities that are really about making better use of health data, to take the leading developing infrastructures for implementing precision medicine, and to be able to shift focus also from towards prevention to tackle both this and also future pandemics, of course. In the strategy, we talk a lot about the future challenges with the development of demography. We talk about pandemics and the underpinning AMR situation. We had, of course, no clue in December that we were so near the situation only a few weeks later. The lessons learned that we've seen from COVID so far is, of course, that it's very clear that we need an even stronger resilience in our society. We need a health resilience and sustainable solutions for our future competitiveness and for future health. Therefore, I cannot stress enough the strength and importance of the huge investment from the Wallenberg Foundation in data-driven life science that we've seen. And we're so pleased for that. That will really enable to lay the great new foundations for life sciences with impact not only for Sweden, but for our global community in this field. With these opening remarks, I look so much forward to being part of the discussion over the day, and look so much forward to your presentations. Thank you so much.
Thank you so much, Jenny. That's a very, very good start of the day. And I would like to encourage everyone who listen to post questions on the questions and answers And now I will invite Olle Kallioniemi, the SciLifeLab Director, to speak about SciLife-KAW COVID-19 National Research Program. Please, Olle.
All right. Thank you so much, Siv, and welcome everybody to this symposium. I'm very happy to have this event today and so many people participating. So what I will do quickly is really like a 2-minute introduction to some aspects of COVID, and then go to the SciLifeLab-KAW overview of what has happened in the last 6 to 9 months, and then at the end say a few words about the future of the program. So just to show the very basic facts where we are now, that pandemic is not going away. We have 500,000 new cases in the world, 5,000 deaths per day, and that is really significant. It's also not going away from Europe. We are seeing a strong second wave, or maybe just the resurgence of the first wave, 200,000 new cases per day and 2,000 deaths per day. This map shows how the kind of a center in Europe is now in Central and Southern Europe. And the situation in the Sweden and Nordics is a lot calmer. Sweden has now taken the lead in the Nordic countries again in terms of the number of cases per million. And noteworthy is also that the percentage of positivity is above the 2% limit that is considered significant. But the situation in Sweden compared to some other countries in Europe is obviously much better. But the yellow color and the red colors here indicate that the pandemic is still on the increase. This has also been a very special time in the sense that the scientific process has been put into a test, that we are typically publishing in a very slow pace, and data comes out of a scientific publication in a year or two. And this has dramatically changed. The journals turn over the papers much more quickly, and people deposit all of their preprints available online. And this has really been considered how this is maybe how we should do science in the future. And today, some 60,000 publications in PubMed. This means that several hundred papers are being published on a daily basis on COVID. I found from the PubMed some 700 from Sweden. And if you look at what these papers are in terms of the nature, obviously a lot is on epidemiology, diagnostics, drugs, and so forth. And we really, when we consider like the program that we are discussing today, have to put the advances in Sweden in perspective with what's happening in the world when so much effort is being put on COVID-19. For instance, obviously everybody is waiting for the vaccines now. So we have hundreds of candidates in the preclinical states and dozens of candidates already in clinical testing. So this will turn yet another important phase in COVID-19 as to how the vaccine is starting to work on a population level. And obviously we are not just having papers, we are having tons of data arising out of these papers. This is just one aspect of the data, meaning the viral sequence evolution that can be traced online in real time, which is really the unique aspect of this. So at SciLifeLab in about mid-March, we started to wake up to the reality that we should do something about COVID-19. So first thing we did was to prioritize the use of the national infrastructure in COVID research, started the process on how to set up testing for virus and engage the research community in a research program. And this has really been a national effort funded largely by the Knut and Alice Wallenberg Foundation, also by SciLifeLab national funding, together with the healthcare hospitals and healthcare system and the hospitals and all universities in Sweden. And I'll say a few words on just introducing these topics because this will be described today in detail. So setting up the testing, Lars Engström will talk about how the KI Pandemic Center for Virus Testing has been launched. Sophia Huber will talk about the high-throughput, high-content serology that has been done at KTA at SciLifeLab and how that has helped to understand the immune response in patients and looking at how durable those responses are. And all these statistics on how many tests have been performed are actually available online in real time through the COVID-19 data portal that Johan Rung will describe later on. So noteworthy to say that there are 350,000 virus tests already done, some 80,000 serology tests. And you kind of see a little bit of an uptick on positivity percentages already in this type of a plot as well. I would also like to say that we really should consider unbiased ways of monitoring the viral load in the society and environment. And this is what the environmental virus profiling group has been working on, looking at viral loads in the wastewater, for instance, and in the environment. And there was a recent picture in Dagens Nyheter where Cecilia Williams was also participating in overlaying this type of testing, which is unbiased and is not influenced by how many people go and get tested. You really get the whole population load, and how that also shows that the virus load is increasing in the population. So then a few words on the research program. So we wanted, with the KAW funding, really quickly set up a national research program on COVID research. We wanted to do it in a manner that best fits from the SciLifeLab perspective. How could we also make use of the infrastructure? And what we did was to launch a public call, got almost 300 applications. And when processing them through, realized how many people are suggesting similar studies, like how to diagnose a virus, how to do serology, and so forth. And we decided that we should not only fund these programs, but make people collaborate within these programs together and assign them into research areas and coordinate the research more. So this is and has been a really unique program in a number of different ways. So I'll just point out 5 points: rapid selection, rapid getting going, asking scientists to collaborate rather than just choosing 1 or 2 that will be funded, with coordination, provide funding for the biobanks in order to address the link to the healthcare, and also support SciLifeLab infrastructure to boost capabilities for COVID research. And then finally create a data portal for sharing the results openly. And this program has now supported all major universities in Sweden, and the different research areas have been assigned a coordinator and a scientific lead, and many of these people will talk about today. Just quickly a few snapshots. So first of all, we considered important that we launched the biobanking initiative, and Mia Philipson will talk about this in a bit more, on how this could get going in April already during the peak infection times. And this has really been important. The data portal that I mentioned earlier, Johan Rung will talk about that later, but this is really a data portal for all research, not just for this program, all COVID-19 research in Sweden. And you'll hear a lot more about this one in a short while. So what I'll do now in the next 1 minute is, what are the next steps? And I think this is really one opportunity for us to kind of discuss this and maybe get the feedback from the entire community on what do you think. One is the testing aspect, pandemic preparedness. How should we promote it in the future and keep up the capabilities? And there are funding opportunities here because government has planned to target support for SciLifeLab for the next 3 years. The research program, how do we continue the research program that we already set up? And should we incorporate new topics into this? And here KAW has provided another 50 million for the next 2 years to continue some aspects as well as to update other areas to it. And we need to make decisions. We are not in such a hurry as we were in the spring. So this will take some time before we launch any actions here. And then the data program, how we can best promote data-driven and computational approaches. And this is where the data-driven life science program that was just launched on Wednesday actually helps us. And if you did not participate in our launch for the data-driven life science, this is really the huge investment from the Wallenberg Foundation for data-driven life science. And one area of this data-driven life science is epidemiology and infection biology, which should help us also to promote this aspect of COVID-19 and pandemic preparedness. And my final point is to say that as part of the COVID-19 effort, we will be starting a seminar series now on topics around these research areas that we will be supporting. And I'm sure we will have these types of events repeating as well in the near future. You will hear more about us, and please feel free to contact us. These are the contact points where you can suggest topics for the research program and any general comments on how we should continue what we have been doing thus far. So thank you very much, and look forward to hearing all of your talks today. Thanks.
Thank you very much, Olli. And we have one quick question. Is the number of published publications for other topics than COVID-19 similarly high, or did it go down because journal focused on COVID publications?
That's a really good question. I don't know. So that will be worth checking out.
Yes, that would be interesting to find out.
But certainly maybe virologists at least have seen a shift from other virology topics to COVID-19 for sure.
And our next speaker is Professor Lars Engström from the Karolinska Institute, and he will speak about the KAW Pandemic Center for Virus Testing. Please, Lars.
Yes. Let's see here. Thank you so much for giving me the opportunity to share what we have been doing for the last 7 months since we started this test center and research center at the Biomedicum at KAW and with SciLifeLab. So my name is Lars Engström. I'm a professor in infectious disease control and also a physician at the hospital side. So we are quite a lot of collaborating between the hospitals, not only in Stockholm, but also in different regions from Sweden. So we can consider us, I think, as a national pandemic center for diagnostic. We are from the beginning a research facility, a center for translational microbiome research. So what we are doing normally is to study the human microbiome in health and disease. And we are very much studying this together with clinical collaborators. So it's a translational research, hopefully leading to clinical applications in the future. So this was our main focus before this happened in the beginning of this year. So then I got an email from my colleague, Jan Albert, asking us about, are there any opportunity for you to help us with RNA extraction? Because they were not able to handle this at the hospital side, and they knew that we had access to some kind of infrastructure for this. And fortunately, we had some of the equipment needed to speed up this and create the high throughput facility. We didn't have any support for the sampling side in the beginning, so we had to invent lots of things from that side, but also moving this into some kind of throughput activity where we have an inactivated area, and then we do the RNA extraction in the robots on the right side, and then we went into the qPCR. So this is a quite straightforward technology or platform that is used all over Sweden and all over the world. The challenge here was, though, to speed up and also increase the capacity. That means to have a high throughput focus on COVID diagnostics. The problem in the beginning of April was that we didn't have access to enough robots here. And thanks to Wallenberg Foundation, we were given 60 million Swedish crowns to set up and start up a COVID testing center at Karolinska and SciLifeLab. This was some kind of situation between Fawlty Towers and the Bond movie, actually. We were really worried about the transport. It was a very special feeling to don't know if the equipment will arrive or not. But at the end, after a couple of hectic days, we had 5 tons of equipment here from Hong Kong. and the aircraft was organized by the Wallenberg Foundation. So everything was very fast. And after a couple of days, we had everything in-house. You've seen these pictures. It was quite busy in the lab. We had to rearrange quite a lot of areas to this. We had to educate many volunteers from SciLifeLab and from KAW, who actually really did this on a voluntary basis to help to set up and speed up the process. The robots were up and running. We had programmers from China coming here, helping us to arrange for this. And we also had this packing center where we pack all the test material that are now sent out to the different regions. And today we have a capacity of 7,000 samples per day. So you can imagine how many packing people we have here. And then we have 10 of these SP960 robots. This is the largest lab in the Northern Europe now for COVID testing. And this means we can use these robots for all kind of not only for diagnostics, but also for sequencing of the virus. And I will come back to that a little bit later. And we also had help with the technicians from China. They were scared to death to come to Sweden, and you can see that here. But fortunately, we have access to the T7 machine on the right side that I will tell you more about when sequencing the virus in a high throughput mode. So after 4 weeks, we were up and running and ready to go here in Stockholm with a capacity of 5,000 samples per day. It was a little bit frustrating during May when we couldn't see any samples coming from the regions, and there was a debate about why this was the case. And I'm quite sure that this debate will continue during the Corona Commission, because it was a little bit frustrating to have this capacity, but very limited number of samples. So what have we achieved then after 7 months here in Stockholm? Well, you heard about the number of tests on this webpage. You have an updated version, Ollie. Today we have sequenced 350,000 or more than that samples. This is the largest lab in Sweden and Northern Europe, as I said. We have more than 16,000 positive cases in the biobanks. So this is a gold mine, of course, for research in the future. And you can also see, as Ollie mentioned, that we can see an increase in the numbers here now. Most of these samples are from the Stockholm area, and you can also see here down that the cumulative test numbers have increased quite dramatically since the end of August, and it increased every day. I think we had 8,000 samples here yesterday. And we can see this also when you look on the curves in Stockholm on the right side here. Here we have the numbers of positive cases. And of course, this is the reason in many cases that we have this kind of problem now. And yesterday, the Public Health Agency banned parties and things and dancing like this, much based on the numbers we see now, unfortunately. So we have 2 tracks. We have one track from here into our lab, and then we go with the 350,000 samples for diagnostics. But we have also, as I said, this biobank. So we have sequenced more than 2,000 whole genome sequencing on the strain level. And thanks to this machine park that we have now with 4 of these DNB-seq 400 machines and this T7 machine, we have a high sequencing capacity in the lab, and we are using that for studying mutations in the virus. And there are 2 options to do this. Using the multiplex PCR-based sequencing on the left side here. Here we only focus on the virus genomes. And this is a high throughput approach with a very, very high sensitivity. And this could also be used for diagnostic purposes. And we have evaluated this technology, and it is more sensitive than the qPCR approach, actually. But this is another approach, the metagenomic sequencing. We have used that one as well, but the throughput is not that high. You have to sequence extremely deep, but you can still use it to get information in the microbiome genomes, but also on the host genome. And we can also do this in a RNA-seq mode. So we can look at different up and down regulated genes when the virus is present or not. So these 2 approaches have been used here. And as I said, we have sequenced 2,000 strains, and 1,600 something have been very good and easy to assemble. You can also see the capacity here. In one run, we can analyze 960 samples. Compared to if you do this in a metagenomic mode, we can just analyze 16 samples at the same time. So we are now focusing on this multiplex PCR-based sequencing approach, since we can do thousands of strains just in a couple of days. And this is very important because we now want to study what is the mutation rate over time in Sweden and study the different clades that these strains belong to. And here is a tree phylogenetic relationship between these different clades. And you can see here on the right side that one mutation can actually define a new clade. And here we have an example from a cell paper, not from our lab, but where you can show that there is a shift here in clades over time. And this could also have an effect on the magnitude of infection. Many people are now discussing if the mutation can make the virus less virulent, but more efficient for spread, or the opposite. And we have an opportunity to study this since we have now 15,000 strains in the biobank. And here are some of the examples. As we just saw, there are two major clades, the 20B and the 20C clade. And you can see that the CTMR strains belong mostly to the 20B clade. But we also have other strains from the 20C and the 20A clade. And if we use this to study where are the viruses coming from, we can see here that there are not so many strains in Sweden up till May that is from the Asian part. There are strains from all over North America, South America, Europe, and so forth. So if you go into this kind of investigation, we can track where the virus comes from. And we can also use this now in our collaboration with epidemiologists to study also within cities, and if there is a certain zip code that is associated with the spread of a certain strain. So this is a very powerful tool, and we hope that we can develop this together with our epidemiology colleagues, but also with other colleagues all over Sweden. And you can see here also that what happened in May or in June was that the clade 2 actually took over here in Sweden compared to in the beginning of or in the springtime here. So this is a way to study the dominance and the frequency of different clades in Sweden. And this is important to study also the spread of disease, where it comes from. So the plans now are that we will look into the mutations and search for the ones that can change the transmission capacity or virulence. And we also work now with the BSL-3 lab here at Biomedicum with in vitro models to study the virulence of the virus. We also want to adapt the nomenclature with higher resolution than the current one, the next strain, and we are working with bioinformaticians from here and China to develop this. We want to have phylogenetic analysis on the Nordic countries, so we are asking for strains from all over the Scandinavian and Nordic countries. And we are now sequencing clinical samples in the fall to see how this develops. We have had quite a lot of interest from media here during the setup of this lab, and it has been a little bit challenging to explain to everyone why we didn't have virus tests in May when everything was up and running. That could be discussed later on in other occasions. This is the team of heroes, Fredrik, Mike, Jessica, and Marika, who have been organizing and all the high throughput capacity here in Stockholm. But it's also good to have support from young colleagues. This is to the researchers at the Karolinska Institute, and we've got very good proposals for how to handle this virus. This is a robot that eats viruses, and here's a treatment issue. You can drink lots of lemonade and then get rid of the virus this way. So this is the fun part of what we are doing. Thank you.
Thank you very much, Lars. It's truly impressive. We have got a number of questions, but because of time constraints, I think we need to move on to the next speaker, which is Sofia Huber, and she will from KTH, and she will speak about high throughput and high content serology testing and research. Please, Sofia.
Thank you so much for giving me the opportunity to discuss what we have done the last 7 months here at the SciLifeLab meeting. And I can't enough stress the fact that SciLifeLab and Knut and Alice Wallenberg's foundation has been instrumental here in order to not only to give us funding, but also to make a network, making us able to work together in a new way that we haven't been able to do before. So I will present some short parts of what we have done when we developed the high throughput, high content serology testing, but also how we have used this on the personnel at Danderyds Hospital. And this started an evening in March when my colleague My Hedhammar called me and said, we need to do something, what can we do? And we, together with Peter Nilsson, discussed what are we able to do and what kind of tools do we already have in the lab. And then we went out to develop this serology method. Sorry for that. As you know, it has been very much discussion about finding antibodies in different persons' blood. And this can be been done in several ways. Either you can look for IgA, IgM, and IgG, but for the long-term immunity, it's IgG that is the most important part. And this can be done either by point-of-care test. You take a sample one at a time, and you can get an answer in 15 minutes. But you can also do this in a lab analysis when we draw a vein blood sample and send it to a lab, and there you can do it in much more high throughput context, either by ELISA or by Luminex. And normally those lab analyses are a bit more secure in what it reports out compared to the point-of-care tests. The difference with our test compared to normal ELISA is that we can do this in a multiplex manner. We can use a number of different color-coded beads where each beads represent a specific antigen. And the antigens are currently made in-house. When we started up this, we used 160 different antigens in order to see what is the best version of the antigens coming from the virus. And each antigen has a specific color code. So then we can mix all these different beads up to 380. We have only used 160 so far. And right now we are only using 5 in our final setup. So we mix the beads with a sample from the patient. And then if there is an antibody binding to a specific antigen, it will bind to the bead. And the instrument can measure what kind of bead it is and thereby know exactly what antigen is on the bead or what part of the virus. And then it also measures how many antibodies are there. And when we run 227 positive samples and measuring the signal coming out from the different beads, you can see that here in this assay, most of the antigens actually give a signal. And what we are doing in deciding if it's a yes or no, we compare the signal with a negative control. And then we demand the signal to be 6 times standard deviation higher than the mean of the negative controls. And when we then transform these signals to yes or no, it will look like this. Some of the antigens actually give a yes on almost all samples, while some of the antigens are not that good in showing if there is any antibodies, or probably there are not so many antibodies in these blood samples. Here we have an individual that actually have not developed any antibodies. But if you look at this picture, it seems to be rather straightforward. You just pick one of these antigens that actually are giving a good signal for most of the samples. And then you run all the samples on this antigen and you will have yes or no. But if you it then makes life a bit more complicated if you compare this with negative control samples. And here you can see out of 462, I'm here showing 166 samples that actually give a background signals, even if the individual cannot have had the disease because the individual or the sample is taken before 2020 in 2019. And if we transform all these signals to a yes or no, you can see that there will be a false positive in every single variant of this antigen. So here, if we use the one that looked best in our As I showed you before, there are a number of people that actually show that they have some kind of antibodies binding to the spike. So in order to, here you can see also the different samples in a plot where you can see that here is a negative controls showing a number of people having antibodies against this spike protein, and so on and so forth for these different 5 that we have in our final assay. So in order to solve this, we decided to go for demanding more signal from more than one antigen in our assay. It has been shown that if we use a fold spike and the C terminal of the nucleocapsid, we get the best signals out. Here we have around 332 different samples, and it's only this guy that we couldn't detect any antibodies that do not have antibodies to these 2. And among 290 different negative samples, we actually have no person having antibodies against both those 2. So this gives us a sensitivity of 99.7 and a specificity of 100%, running 2090 negative samples and 332 positive samples. And this assay has been used for evaluating or looking at personnel at Danderyds Hospital. And here we have analyzed 2,149 different employees. We took blood from these people in end April, beginning of May. And it turned out that 90% of these employees actually had antibodies against the virus. You couldn't see any difference between women and men. And about 9% of these reported no symptoms at all, and still they show antibodies. And we could see, as many have reported, that loss of smell and taste are the most significant symptoms when it comes to this disease. Moreover, what is very important to notice, we could see that there were a very big difference between those people working at the hospital having no patient contact, where had the same spread of the disease as the normal population in Stockholm at that time, while those working with patients actually have a high degree of infection. And also when we divided them into no COVID patient contact and COVID patient contact, we could see that those people that work with patient where they thought wasn't infected actually also get a high infection rate. And when we divide them in different departments, dealing with patients in different ways, we could see that there were a large difference depending on where you were working at the hospital. Now in the fall, we have looked at these personnel once again. We were really happy because 92% of the full more than 2,000 persons actually come and gave blood again. And among those that had antibodies, 99% participated. And what we could see now, we are still, of course, looking into this data from different angles, but we could see that 20% of those that actually were positive in our assay before the summer didn't turn out positive now. We could still see antibodies, of course, but the levels were really low. We could also see that 5%, about 100 people, have had the disease since the end of May. So important conclusion from this work is that we have developed a highly stringent and high-throughput SARS-CoV assay. And we use 2 antigens in order to ensure high specificity and also high sensitivity. And I think that the high sensitivity we are able to keep here is due to these 2 antigens as well. More than 80,000 samples, as Olli said in the beginning, have been analyzed, and 58,000 has been individually reported out to people for them to know if they have antibodies or not. And that I can mention that we actually find antibodies in persons that do not report it out positive in the normal test that is run at Landstinget or 1177 right now. Also, I would like to stress that personnel with patient contact at Danderyd were at much higher risk to get infected compared to others. So I think we need more stringent measures to increase the safety for healthcare personnel during fall now, when we see that everything is coming back again. And we could also see that the duration of the antibodies are limited, and also that those are dependent on the severity of the infection. So this is my last slide. I would like to acknowledge, of course, TG Lab, Hedemar Lab, that has been part of this, Nilsson Lab, and the autoimmunity facility at SciLifeLab that has been instrumental in running all the samples, and Thulin Lab that has worked with with the personnel and also sample them, and we have a really good collaboration. Of course, Wallenberg, Knut and Alice Wallenberg's foundation, as well as many other that has been giving us money to drive this project through. Thank you so much for listening.
Thank you so much, Sofia. This is truly excellent work. And we now need to go on with Anna Shekeli from Uppsala University, who will speak about environmental biobanking and virus profiling. Please, Anna.
Thank you. Let me share my screen. So, thank you very much. I'm here to talk in the name of the environmental virus profiling research area of SciLifeLab's COVID action, which Olli already introduced a little bit. But before I jump into our results, I want to talk a little bit about why it's interesting to do environmental virus profiling. Already at the beginning of the pandemic back in March, studies showing how long persists coronavirus in different environmental habitats like surfaces of the built environment were published. We learned for how long we should not touch a plastic surface previously touched by an infected person. But beside these coronaviruses that were infectious, we learned that the genetic material of the virus can be detected even longer. For example, one of the first long-term recovery of SARS-CoV-2 RNA came from the Diamond Princess, where the virus could be detected in the cabins of infected people even 17 days after they left the ship. What does this mean for research? Well, in one aspect, it means that our environment is full with the virus. It's in the air, it's on surfaces, it's on our body. This means that all these environments can mean a risk of infection, and the surveillance of these environments from an infection risk estimate perspective is important. However, as environmental scientists noticed already in the beginning, the profiling of this kind of environmental space, this built environment that surrounds us, allows us to analyze the virus in a much broader perspective. We can analyze the virus not only through looking for what virus is in the most severe symptomatic people, but we can look into it on a population level. And it's very important to collect samples from this kind of population level sample because they can represent unrepeatable imprints of the genetic status of the virus at given space and time. This idea became even more important when the first report came out that the virus, although it's a respiratory infection, or that's what we thought in the beginning, it's also present in great numbers in fecal samples. Actually, it was reported that it can be detected for much longer in fecal samples than in respiratory samples, and that up to 10 on the 8th copies per gram of fecal sample can be enumerated. Soon after these results came the first detection of the novel coronavirus in wastewater. It was first detected in the Netherlands, in the wastewater treatment plant of the Schiphol airport. And then this already rang the bell to many scientists involved in wastewater analysis that this can provide a great opportunity for wastewater-based epidemiology of the SARS-CoV-2. But what is exactly wastewater-based epidemiology? Well, wastewater-based epidemiology is a population-level surveillance method. What does it mean? It means that everyone who is connected to a wastewater treatment plant donates an anonymous samples every day to the wastewater sample. We can't avoid this unless we have our own water treatment sample. And then by sampling the wastewater at the incoming flow of a treatment sample, we can basically test the whole population of a given municipality. Then we analyze these samples, and it has been shown that this kind of analysis and detection of SARS-CoV-2 can be a leading indicator of infection surges to 4-10 days. What does it mean? It means that if we monitor the wastewater, we can detect the virus ahead, and we can adjust the different MPIs, the non-pharmaceutical interventions. Furthermore, by collecting the wastewater samples and biobanking them, storing them for the future, we can provide a sample, a population level sample for future researches that can't be reproduced again. So, as Ulle already introduced our research area a bit, we all applied for funding from SciLifeLab to do this kind of environmental virus profiling. We are 4 groups, and we will put together to collaborate in this project. So 3 of our groups work on wastewater-based epidemiology, and 1 group, led by KAW from SLU, works with surfaces, primarily with surfaces in the Stockholm underground. He has been working sampling the subways of Stockholm already before within the MetaSub consortium, but he also did 3 samplings with his group in April, May, and June in 2020 and collected unrepeatable samples from Stockholm wastewater subway stations. They collected swabs from different surfaces, and many already have been shown to be positive, like elevator buttons and handrails. Furthermore, together with the MetaSub consortium, they perform metagenomic sequencing of these swabs. For the other groups, the first group working on wastewater-based epidemiology is located at KTH and is led by Zeynep Setigalugurul. Their group aims to focus on Stockholm wastewater. So they started to collect weekly samples from Stockholm from the beginning of April. They sample 3 different inlets in Stockholm and cover almost 2 million people. Basically, they do a corona test for 2 million people every week. Furthermore, her group has been very active in method development, and they are collaborating and have early samples of the pandemic from Italy, Catalonia, and the Netherlands, and have been analyzing those. Zeynep works together with many people at SciLifeLab, Cecilia Williams already mentioned Baruli, also at KTH campus and internationally. And on this graph, we can see how the level of SARS-CoV-2 genetic material in the wastewater followed the epidemic curve in Stockholm, according to the weekly cases. The next group is located at SLU and is led by Maja Malmberg. Maja Malmberg is a veterinarian virologist, and she works together with Mette Myrmel, who's a wastewater virologist in the Norwegian Veterinarian School. They recently started a weekly sampling of wastewater treatment plants of Umeå and Örebro. Furthermore, they provided us an indispensable tool to optimize our methods, as they, through the veterinarian context, they could provide us with a bovine coronavirus, which is a beta coronavirus, which is very good because it's very similar to the current SARS-CoV-2 virus. And we could use this virus to optimize all our virus concentration, extraction, recovery, and similar methods. So, and finally comes our group at Uppsala University, led by myself. We also have been involved in many different projects in this less than 6 months since we started our work. For example, we participated in an umbrella study led by the Norman Network, which is a network it's an international network for monitoring emerging environmental substances, mainly led by chemists who now realized, and this chemist has been working with wastewater to assess public health for a long time through analysis of illicit drugs and pharmaceuticals and others. But now we did a cross-section analysis of 9 different cities and also followed very well the epidemic curves. We also worked together with Uppsala Wastewater and Uppsala Vatten, and recently we started to work with the Crash COVID initiative. We informed the public weekly about the wastewater, the SARS-CoV-2 levels in Uppsala wastewater. And we are starting a project where we collected samples from different holiday destinations in Sweden, where there was a big fear that people going on holidays will bring the wastewater, such as Kalmar and Århus. I also work together with many people, both at Uppsala University and many other Swedish higher educational institutes. All of our groups are dedicated to provide our data to the public because we think this is a very good tool to predict the searches. So we have a good contact with the media, and when we see something happening, we want to have it out to the public. Our research area also works together to overcome the challenges that the analysis of environmental samples mean. What are these challenges? Well, compared to the human samples, environmental samples have very low copy numbers. So we are very much working at the detection limit of the qPCR assays. Furthermore, environmental samples are not the same, are not like human samples, which have optimized analytical pipelines. Environmental samples vary and has a lot of other things. So just think of wastewater. And these things all inhibit the enzymatic reactions we perform. So to overcome this inhibition is a big issue. Also, it's not easy to get samples. So we are talking about big amounts of water, frozen, logistic, and so on. And as you have seen, more and more commons want to have this kind of analysis. So we have to develop further to make it more high throughput, our methods. So accordingly, one of our first collaborations within the research area was to optimize the virus concentration methods. As I mentioned, we use this beta coronavirus of bovines. And this already led to a published paper. We are currently planning and very soon running our first pilot about the sequencing of these environmental samples, because as Olli mentioned before, we can also get sequence data from the virus from environmental samples. But because of all these challenges, it's not so obvious how to do it. But soon we will have the first results about these too. And
thank you so much.
Yeah, I'm at the last slide. And we started biobanking samples to not run to a situation as we are now, that we are in an unrepeatable, we can't go, we didn't have samples to look what was the situation in the wastewater back in January or February. So I think this is a very important last point that we do. Thank you.
Thank you, Anna. That's really fantastic work you have done and so important. And we get a lot of questions, and I would really like to encourage all of the panelists and speakers to go into and maybe answer the questions directly in the questions and answers, because now we are a little bit short of time. And we will continue with Johan Rung from the SciLifeLab Data Center, who will speak about the National COVID-19 Data Portal. Please, Johan.
Yes, so let's see. Here we go. So, can you see my slides? Good. So, I will tell you about the data portal that we've set up for the COVID-19 research in Sweden. And as you've seen, I mean, now in this whole epidemic, we've seen data sharing to a point level that we haven't really seen before. Globally, data is shared to a really large degree and is made available very openly. And something that I would like also to stress is that it's important to make data available machine readable. Much of these analyses that you see are only made possible by data being made available on a machine readable way. So why should we share data really, if you have collected a lot of data? Why do it? Well, first of all, sharing data gives transparency into the research process. And this is something, if you have the ability to know what's actually been done, to see the methods behind it, the data that's been used, this gives trust into the research process itself. Reproducibility. If you have access to the data and if you have access to the methods, it's possible for another researcher to reproduce, hopefully. If you cannot reproduce, then you have a problem. And in order to get reproducibility, you need to have access both to the data, but also to the methods, to the code and various things so that you can actually have an openness into what's done. Sometimes you're required to share the data. It could be because of the law. There are legal reasons why you need to share the data sometimes. Sometimes funders will require you to share the data in order as a condition of the funding itself. And of course, as probably most of you know, that today, if you want to publish a paper, you need to make data accessible. Of course, it's also the right thing to do. This is data that is funded by public money. Some of this data comes from samples that have been donated by patients for the purpose of researcher. And if we have an obligation to make the best possible, the maximum impact of what's being collected. And of course, for researchers, what really matters very often is citations. And it's actually been shown that if you publish your data, your study will get more citations. That's important to remember. So what happened in May, and actually already in April, was that on the European level, the European Commission decided that they wanted to support data sharing within Europe. And this task was given to the EMBL-EBI, the European Bioinformatics Institute in Cambridge. They have already around 300 petabytes of data and have archives most major data types. The European Open Science Cloud is an infrastructure where European countries can provide services for other users in Europe. And Elixir is an infrastructure for European bioinformatics. So they set up this European data portal where you can access and submit data into the EBI archives and see how this is being collected in real time. So in Sweden, the government early on gave Vetenskapsrådet an assignment to coordinate this work for data sharing in the country. And Vetenskapsrådet asked SciLifeLab to set up and develop and maintain the Swedish COVID-19 data portal. So we got this one up in early June, and Sweden was actually the first country in Europe to set up a national portal. So now this is operated through SciLifeLab and through NBIS, the National Bioinformatics Infrastructure. So what we have here is a portal. What is the portal in reality? I mean, it's connected to the European data portal to access the data over there. We have services for specific data types, and we have services that are other kind of resources and services for data scientists in Sweden. And this is accessible basically for anyone in the country, for the public, for the data producing platforms, life science researchers. We have healthcare data. Data scientists can use this to mine data, and also for industry. So it's connected, like I said, to the European data portal. On the European side, there's data for virus sequences, host sequences. We have protein data, expression data, and a lot of other type of data accessible. Right now, the European portal gets 3 million web requests in total so far. There's around 130,000 data records and hosted at the EBI. So this is the major database part of the portal. And in Sweden, what we have are support for different types of data types. We have various types of services. We have bioinformatics support. We have a general data repository where you can store data. We have support for text and literature mining, data submission support, and data management plans. But we also have data dashboards for Sweden. We have news about the Swedish COVID-19 research programs, and we have highlights that we show. So in this one, I want to highlight a few of these pages within the portal. One side is the guidelines that we have for researchers. If you're working on a specific data type, how can you work in order to submit your data and make it really open, machine readable, and what should you do with this data? We have news. We pick up publications where we have Swedish researchers, and we bring them up and talk specifically about the data sharing part of these studies. We have the data that you've already seen. Ollie was showing from the virus sequencing at the Engstrand lab and the serology test at Peter Nilsson and Sophia Huber's lab. We have curated records. These are manually curated records from publications coming from Sweden, and how you can access specific data from these publications. And of course, now it's important to generate for your research. Very often is required that you have data management plans. And we have a specific tool and guidelines and support for how to produce your own data management plans. This is also accessible through the portal. So far we had around 5,600 unique visitors and around 30,000 page views, mostly from Sweden. Most focus is coming on the health data, the interest in the statistics, of course, but also for genomics and transcriptomics, how to handle that type of data. And there's a lot of people behind this. I would like to thank SciLifeLab and VR for funding this. And of course, the main development side behind this is Phil Ewells, Per Kraulis, Arnold Kochari, Linus Östberg, and Sebastian Di Lorenzo have contributed to the portal itself. But we also have the coordination and data management side with Hanna Kultima, Niklas Jareborg, Yvonne Kallberg, Elin Kronander, Parul Tebatia, and Katarina Öfors-Stark. So thank you very much.
Thank you very much, Johan. This is so important. And now we will not have a break. We just continue the program. And the next speaker is Mia Philipsson from Uppsala University. And she will speak about national biobanking for COVID-19 research. Please, Mia.
Can you see my screen?
No.
Hey, can you see my screen?
No, only you.
Oh, I'm sorry. You don't see this at all? No.
No.
Okay.
No. Yes, it's fine.
So thank you. I will talk about, So I will talk about the biobanks that has been collected in this effort from enabled by Knut and Alice Wallenberg Foundation, as well as the SciLifeLab. These biobanks were started to be collected already in April during the peak of infection, which is important because it has generated so much information already, thanks to the scientists that have been studying the different patient samples collected at these different geographically spread out sites in Sweden. And we believe that this geographic spread is important because the disease is spreading at different time points. And as you can see, if we start in Umeå, 150 patients with mild to severe COVID-19 disease have been collected. And I know that papers are already starting to come out from these patients. In Uppsala, 125 patients from the intensive care units have been collected, and several publications of these critically ill persons have been published. At Danderyd Hospital, the community study, which I will come back to and which was already mentioned by Sophia Huber, includes 180 patients with moderate to severe disease, as well as half of the personnel working at the hospital. More than 2,000 persons have been sampled. In Örebro, Martin Sundqvist has been collected samples from blood donors and will be collecting samples also from pregnant women. In Gothenburg, 2 different biobanks have been collected. One is collecting 420 asymptomatic individuals with thereby extremely mild disease, and another one is collecting 700 patients, mild to severe disease. And I know that they have been studying, for instance, neurologic implications by the COVID-19 disease. And in Skåne, we have one population study sampling more than 100,000 individuals seeking healthcare due to upper respiratory tract infections. And another one where 60 patients admitted to the emergency department are sampled. And in the population study, several publications are in the pipeline, and I know of one very interesting approach where they will be using the MAX4 in Lund to study receptor virus interactions and how these interactions could be inhibited. So I will briefly describe some of the different samples that are being collected, and this is some of them are collected at all different biobanks, and some are specifically collected in certain places. And this is also a collaborative effort with the Sweden Biobank. And the same samples have to be collected at the different spots in order for us to be able to compare the different disease progression at different geographic sites. So if I return to how the Danderyd study, this Danderyd Community Biobank has been designed, and I will pick this specifically because I was a bit involved in sending personnel as well as test tubes and tube racks to the hospital as they were running out when this biobank was being constructed. So this biobank consists of, as I said, 180 moderate to severe patients, as well as half of the healthcare workers at the hospital. And the healthcare workers have been using an app on their mobile phone to report the symptoms and the exposure, as well as where they live. And they are sampled every 4 months. So we just had them in for the first follow-up, and then assessed for the COVID-19 symptoms and so on. And different parts of this biobank has yielded different results so far, and some of them were described by Sofia Huber already, as this biobank was utilized when designing or developing the antibody test that she described. And in April-May, 90% of the healthcare workers had antibodies towards this virus, which should be compared to 7% in the Stockholm region, demonstrating that we need to protect the healthcare workers so that they are not infected at the workplace, and so that they not spread the virus when they go home from work. So in the follow-up study now in August, we found that 20% of these workers had antibodies, which could be compared to 16% in the Stockholm region of people that have felt sick and sampled themselves or tested themselves for antibodies. And importantly, more than 90% of the healthcare workers showed up for this follow-up, making this a truly unique sample collections that we can use that we can get so much information from in the future. Now, in collaboration with Uppsala University as well as Karolinska Institute, the T-cell immune response to this infection is also being investigated. In another arm of the biobank, prognostic biomarkers are being assessed in these patients, which is important for patient stratification when they are admitted to the hospital, so that the right care is prepared for the different patients. And I know that many papers are already coming out here, together with KTH, Uppsala University, University of North Carolina, and University of Groningen in Holland. This was just examples of what has been coming out from the biobanks together with the scientific programs also sponsored by the Wallenberg Foundation and SciLifeLab. And I think this is of great importance for us to fight this pandemic. So thank you.
Thank you very much, Mia. We have a question here. What kind of samples are collected? Are there any tissue samples collected?
Yes, there are also biopsies being collected at some of these, biobanks. I tried to show you the name of Jeanette Lundblad Magnusson, who is the coordinator of this biobank effort. And it's if you want to know more about the different biobanks, I think it's a great idea to communicate with her.
Thanks a lot, Mia. And then we go on in the program with Petter Brodin from the Karolinska Institute, who will speak about host response to COVID-19 infection. Please, Petter.
Thank you. Can you see my slides now?
Yes.
Is it on presenter's view? No.
No.
Now it is, right?
No. Yes.
Great, thanks so much. I'm going to talk briefly about some of the work we're doing in my team together with collaborators looking at the immune response and the immune system in patients with COVID-19. So me and my team work in the space called systems immunology, which basically means that we are interested in how the immune cells in the blood and the tissues regulate one another, how they work together, rather than just focusing on individual cells and how they function and so on. And so that's what I mean when I talk about systems immunology. This is enabled by a number of technological advances in the last decade or so, and I am directing a facility at SciLifeLab for mass cytometry, which is one of these technologies that we apply. So the logic in everything we do is that we try to measure as much as possible of the immune system. In cases of COVID-19, we follow the blood samples of patients. We measure plasma proteins and gene expression and all of the white blood cells that are present in the blood by mass cytometry. And then by following trends and changes over time, we can infer biological sort of changes that explain, for example, patients with different presentations that can explain responses to treatment and so on. And by this logic, we are currently investigating how this same virus can give rise to such variable presentations, both very mild disease in the majority of patients infected, but also very severe and even lethal COVID-19 in some cases, comparing those. We also have mapped the newly described hyperinflammatory syndrome in children, which is called MIS-C, and I'll talk a little bit about that. And more recently, we have gotten involved together with collaborators looking into the long-term COVID presentations and what might mediate that. So early in March, immunological colleagues of mine and pediatricians such as myself and also other clinicians realized that we need to work together if this is going to be fruitful. And so we formed this network, the COVID Human Genetic Effort, where I am the Swedish coordinator. And here I'm showing you all the sequencing hubs involved in this consortium. We meet every Monday at 2 p.m. Sweden time and talk for 1 hour about our scientific progress, which has been incredibly inspiring. And the logic behind this initially was to try to identify patients with rare presentations, such as previously healthy children and young adults presenting with life-threatening COVID-19, such as, and looking basically for underlying immunodeficiencies in such patients to try to understand what are the host factors that are really necessary to fight this virus. We're also interested in identifying rare patients that might be resistant to infection. For example, family members of patients infected that for some reason do not get infected. Those are very difficult to find, but we're still looking for such patients. And as this pandemic sort of rolled on, we have taken on additional challenges in this network, such as most recently the long COVID and and also the new presentation of hyperinflammatory post-infectious disease in children. So we published the first two papers from this network a few weeks ago in Science, describing two basically phenocopies that explain up to 14% of young, previously healthy individuals with life-threatening COVID-19. One mediated by autoantibodies to type 1 interferons, and the others genetic inborn errors of type 1 interferon immunity, really stressing the importance of type 1 interferons in fighting this virus. In my own group, together with colleagues in Finland, we were able to get samples from patients admitted to the ICU quite early in the spring at the University Hospital in Helsinki. And we applied these methods for systems level immunomonitoring, and we monitored patients over time. And what we found was pretty intriguing, I think, in this publication from a month or so ago. And for example, we identified in these patients that right before patients developed acute respiratory distress syndrome, which is the life-threatening hyperinflammation of the lung, there was an expansion of lung-homing eosinophils, as well as monocytes expressing the fractalkine receptor. This was pretty intriguing, and we think this has these play an important role in the development of acute respiratory distress syndrome. And as many of you know, when cytokine storms occur in the lung, such as in this image here, many different cytokines are involved, and it's probably very difficult to inhibit using targeted therapies only inhibiting, let's say, IL-6, for example, because there are so many other mediators involved. And this has also been shown in recent trials where IL-6 inhibition has been a little bit disappointing. So together with the Swedish biotech company Cancera, we've taken a different approach based on our finding that fractalkine is a homing receptor that might mediate trafficking of immune cells from the blood into the lungs. And so we have an ongoing placebo-controlled, randomized controlled trial at St. Göran's Hospital, ongoing right now, using a fractalkine receptor inhibitor that Cancera has to try to prevent lung hyperinflammation instead of treating it once it arises. And so results of that should be coming out rather soon, I hope. Then as a pediatrician, I started getting alarms from my colleagues in Italy and the UK in April, where they mentioned that these types of children that we normally see and that we recognize as Kawasaki disease patients were coming in at about 30 times higher frequency than normal, which made us quite worried. And we also saw that these children presented with a disease that was quite different from the normal Kawasaki disease. And so efforts began to try to unravel the immunology of this condition, which is now called MIS-C, or multisystem inflammatory syndrome in children with COVID-19. So me and my team worked hard to try to profile these patients using our systems immunology methods. And we published the first report in Cell on September 6th, describing basically the immunology of MIS-C in relation to Kawasaki disease, as well as mild SARS-CoV-2 infection in children. And since this is published, I'm not going to go through all the details, but I want to show a few sort of glimpses of what we found. First and foremost, we can see that the hyperinflammatory state in these children differs significantly from adults with severe COVID-19. Even though both of them have signs of hyperinflammation, the types of inflammatory mediators is completely different. They're also different from classical Kawasaki disease, so we can show here that these are different conditions. We profiled the serological responses to common cold coronaviruses, as well as 1,200 other viruses known to infect human cells using a phage display library that we have in our lab here at SciLifeLab. And we find that all of these MIS-C patients develop antibodies to the SARS-CoV-2 virus. They are relatively comparable to children with mild SARS-CoV-2 infection. But one thing that stood out was that in contrast to all the other children we looked at, these patients that developed MIS-C lacked IgG antibodies to common cold coronaviruses. Whether that has a pathological role or not, we don't know yet, but we're looking at that. Finally, the hyperinflammation in MIS-C, as well as Kawasaki disease, has been proposed to be, it's likely that it's an autoimmune response triggered by the viral infection. It occurs about 1 month after the initial infection. And we looked using whole proteome arrays for autoantibodies. And what we found several candidates, and one pretty intriguing group of proteins are these casein kinases. And the reason this is intriguing is because the signal is pretty specific to MIS-C. We don't see antibodies in other conditions at the same levels. And more importantly, a previous report from a San Francisco-based group showed that these particular casein kinases are strongly induced in cells infected with the SARS-CoV-2 virus, suggesting that these cells upregulate these proteins, the immune response to the virus cross-reacts with these proteins, and that might mediate this hyperinflammatory syndrome. So these were just some glimpses of the systems immunology work we're doing. We are very grateful for the funding from this SciLifeLab COVID-19 research program, as well as private donations. Thank you very much.
Thank you very much, Peter. And we have a question here. What are the treatment options available for MIS-C?
Well, we give a cocktail of various immunomodulatory drugs, both targeted specific cytokines, steroids, and IVIG. But that's it's a very complex issue that has to be done by specialists in pediatric immunology.
Thanks a lot. And then we continue with Emma Lundberg, who will speak about cellular targets and biology of COVID-19 infection. Please, Emma.
Hi, everyone. It's a pleasure to talk here today. And I'm talking on behalf of the team that you can see on this slide, an amazing team of researchers, Charlotte, Päivi, Brinton, Anna-Lena, and Ali. And I'm going to talk about the cellular targets and biology of COVID-19 infection. And the aim of our research area is to identify novel host targets and repurpose existing drugs. And as you've heard, the virus hijacks the machinery of the human cells to produce more viruses. And by better understanding this, we hope that we can identify novel potential targets. You can see here on this slide, for example, existing drugs where they hit on this infection trajectory, replication life cycle. But we hypothesize that by better understanding which proteins are either upregulated, they might be beneficial for the virus, or the proteins that are downregulated would interfere with the viral production. But we also want to identify the proteins that translocate in the cell so that the virus takes proteins, bring them to another part in the cell, and basically recruit their activity for their own purposes. So in this program, we want to identify all those proteins, and we have a pretty unique opportunity to do this here at SciLifeLab. So we set up this project where we first do single cell spatial mapping of the SARS-CoV-2 interacting human proteins. And based on the output of this study, we will do targeted drug screens with the hits from number 1 as a readout. And this is only made possible because of the synergy from SciLifeLab, so the Human Protein Atlas antibodies, the BSL-3 labs, the drug screening facilities, the cell profiling facility, and so on. So we started by looking at literature, and as Johan talked about, it's important with open and transparent science, and thanks to preprints, we were able to find a list of 700 human proteins that were either shown to interact with SARS-CoV-2 or other coronaviruses, and we decided to include these in our study. And this was really important because we could get started very early summer with this screening.
Emma, it has been pointed out that the slides are not full screen.
Okay, are you seeing my presenter mode or?
Yes.
Ah, okay, sorry. Now, is it good now?
Yes, it's fine.
Yes, okay. Thanks for pointing that out. Yes, so we then get started with establishing this assay. And the first part then starts with infection of cells. And then we do this automated immunostaining where we visualize the host proteins, high-throughput imaging, and computational image analysis. And at the endpoint here, we will end up with a ranked list of host proteins that we think are interesting. And then we do a target-based repurposing drug screen. And without going into the details here, I just want to point out that this is really a massive screen. We're looking at 700 different human proteins here as targets. And then for the drug screen, we will do, I think, 8,000 samples per target. So it's really big data being generated here, and we're very dependent on this data platform for computational image analysis as well. So to show you what it looks like, here we see some cells. We're using Vero E6, so this is monkey cells, to get started quickly. And the nuclei are shown in blue. And the virus is visualized with an antibody that is red here. So you can see that about half of the cells are infected and half of the cells are not. And this is on purpose because we have positive and negative controls in the same image. So we can do a segmentation, computational image analysis based on these images. But for this presentation, I will just show you some examples of what we're looking for. So to the left here, you can see an example of a protein that is upregulated in infected cells. So you can see if I turn off the red, you can see that in the red cells, the protein of interest is expressed at higher levels. Whereas this protein, you can see that it's not expressed anymore in the infected cells. So altogether, we could find 178 host proteins that were either upregulated or downregulated, and most of them were upregulated. And then we also looked at these spatial translocations. It might not seem very dramatic if you're not used to looking at these images, but this protein, for example, it's only in the nucleus in non-infected cells, but we also find it in the Golgi apparatus in all the infected cells. And this is actually very dramatic to the cell. To have a protein that is never supposed to be in that compartment all of a sudden come in there, it can mess up a lot of things. So, and altogether, we were surprised by this high number. We found 84 proteins to show such translocations. We see certain trends here, for example, that proteins that are normally not on the plasma membrane tend to go there, and they might be important for forming the shape of the cell, as Petra talked about, or viral entry or virus exit. We also see that proteins from the secretory pathway go to the nucleus, where they might have gene regulatory functions. Mitochondrial and microtubule accessory proteins also seem to leave their original location and go all over the cell. So we're right now at the point where we've, in addition to these known targets that existing drugs are targeting, have identified another range of potential host targets of relevance. And we're now starting to build this drug screening assay with a targeted spatial readout. So you can say that we're about halfway in this project, and we're really excited about getting started with this drug screening. So what I do want to say is that the top part here is what has been funded with this SciLifeLab COVID-19 Wallenberg grants. But of course, we hope to be able to continue this project with candidate drugs that we hopefully find. And I also want to say that behind all this work, there's been a lot of technology and methodology optimization throughout every single step here. But what I'd like to emphasize is that this is a really unique platform. I don't know of any other labs in the world that are equipped to do all of this targeted phenotypic screening that we could think of using for identification of host targets and in other diseases, and also repurposing of drugs in other diseases. And one important thing that I would also like to highlight is that Marju Pumalainen at KI has set up the safety level 3 lab, which means that she's the one in the middle here, means that they can work with SARS-CoV-2 viruses, and they can infect cells for us, and we can use these for validation in human cells and good cell models. So this is really important for all this work. And with that, I would like to end my talk and thank everyone that has been involved. It's really a fun project and team science at its best. And I would also like to highlight Jay, who's been doing a lot of the work with imaging to date. Thank you.
Thank you, Emma. That's really interesting. And a question for you. How would you explain the change in subcellular localization? Is it because of a general bad state of the cells or change of the targeting signals in the proteins?
I think it's because of interactions with the viruses. So different viral proteins will go to different parts of the cell, and they might hijack proteins and bring them to other compartments. So I think, I don't know for sure, but I think it's mainly interaction patterns.
Okay, thank you. And then the next speaker is Christian Sandberg from Uppsala University. and he will speak about drug discovery and studies of drug targets for COVID-19. Please, Christian.
Thank you. I'm trying to share my screen here. So you see this in presentation mode now?
Yes, it's fine.
Thank you. My name is Christian Sandberg. I'm the co-director for the Drug Discovery and Development Platform at SciLifeLab, and I will talk about drug discovery and studies of drug targets for COVID-19 performed at SciLifeLab. So what is drug discovery? Drug discovery is actually a process of how you apply various technologies within different research areas to develop a drug. So how can SciLifeLab contribute in drug discovery? First, build awareness of the need for academic science in drug discovery. Secondly, secure quality of generated data. Thirdly, frontline technologies are available at SciLifeLab, use them. And fourthly, we have one infrastructure, the Drug Discovery and Development Platform, with competence and capacity totally dedicated to drug discovery. SciLifeLab and Knut and Alice Wallenberg Foundation is supporting 10 projects involving 50 research groups for research in drug discovery and repurposing of drugs. In support of these projects, a major current focus is to build a chemocentric database to allow key information on compounds, assays, and generated data to be shared with the scientific community. And several of the platforms at SciLifeLab are involved in this work. This is an overview of ongoing projects for drug discovery at SciLifeLab. These projects could be divided into 3 groups. The first is actually what Emma talked about here, phenotypic assays and target identification. Target identification is when new biology is discovered and assays, often cellular phenotypic assays, are developed to study this biology. And I believe that Emma and her co-workers have 12 projects in this phase, covering in principle all interaction and interplay between the virus and the human host cell, depicted here. These novel assays can, as Emma said, be used to screen compound collections in phenotypic assays, including known drugs, but also experimental compounds that eventually could be developed into drugs. And we are currently running 3 projects in this phase. If the molecular drug target can be identified and defined, dedicated drug discovery projects can be launched with the aim to generate prototype drugs. We have 6 projects in this group right now. Concerning these projects, we have 3 projects focusing on the main protease of SARS-CoV-2, which is an enzyme that cleaves the polyprotein produced by the viral genome into a multitude of functional proteins. It's many projects here, and I encourage you to look at the SciLifeLab page to learn more about these projects. So why are 3 projects interested in the main protease? First of all, the main protease has been shown to be absolutely required for replication and amplification of the virus. Secondly, this enzyme is very different compared to human proteases, which means that there is a higher probability that future drugs will not interact with human proteins and cause side effects. Furthermore, and at the same time, the main protease is found in all coronaviruses, which suggests that main protease inhibitors could be useful also for future pandemics. Thirdly, the main protease is well defined. This is illustrated in this slide, where our close collaborators, Frank von Delft and Martin Walsh, published a paper in Nature earlier this month, describing how small fragments are binding to the catalytic site of the protein, but also identified a binding site in the interface between the 2 parts of this homodimer, suggesting another allosteric site of the protein. Pfizer has a main protease inhibitor in the clinical phase for COVID-19. interestingly, this treatment appears to be synergistic with remdesivir and enhances the antiviral efficacy of remdesivir in cell cultures. But as remdesivir, this drug has to be given intravenously. So is there a need for a new main protease inhibitor? Dennis Liotta at MRI Institute phrased it very nice in a statement on the web. The pressing need now continues to be the development of orally available agents which can be used broadly to treat COVID-19 infected individuals and to prophylax uninfected individuals who are at the risk of becoming infected. Hence, the use of the Pfizer drug is hampered by its intravenous route of administration. So our goal is therefore to generate new prototype drugs suitable for oral uptake. We have also concluded that new chemistry is required because there are no good chemical starting points to develop this type of drug. So how to find new chemistry for the main protease? We have chosen to fingerprint the chemical space to allow for machine learning. At this stage, we are setting up a workflow using 3 different approaches for drug discovery, combined with quality controlled assays and iterative chemistry to generate high quality data. We use computer-aided drug design that compares the structural information of the protein with structural and chemophysiological properties of compounds from sources containing more than 800 million compounds to select approximately 100 to 200 compounds per round to be tested in our assays. We are also identifying small molecular fragments from collection containing a few thousand compounds using biophysical assays. And this information is used to refine the computer algorithms used for in silico screening. We are also using DNA-encoded chemical libraries containing billions of compounds to select a handful of binders, and their properties are also used to refine the computer algorithms. As a note, SciLifeLab is the only academic discovery center in the world that has developed our own DNA-encoded chemical library and is providing these types of service to the academic scientists. Data from these approaches can be used as fingerprints in the chemical space to describe how a functional inhibitor should look like, and then machine learning can be applied to further refine information collected from these 2 approaches and data from the assays below. Yes, we have found new chemistry that we are currently refining in our assays with iterative chemistry. And we hope to start machine learning next year to speed up our work and to dig deeper into the bank of information that we are building and exploring much more of the chemical space. And of course, data will be shared on the SciLifeLab portal. And just showing this slide, we have a huge collaborative network also for these collaborations. I stop there.
Thank you, Christian. And a question that has come up is why is drug discovery important for university scientists when big pharma invests so much in drug development?
It's a good question, but sorry, I have to blow my nose. It is important to understand that healthcare and big pharma are increasingly dependent on academic research and spin-outs from universities as a source of innovative drugs through the discovery of prototype drugs. The pharma industry is very, very good at the development phase when you have a candidate drug that you are going to be tested in the clinics. So I strongly believe that if you want a healthy academic drug, a healthy academic drug discovery branch is essentially for a functional life science sector that can attract and work with pharma industry.
Thank you. And then we continue in the program. And now we have invited Åsa Kristoffersson Hedlund, who is the chair of the Svenska Covid-föreningen. And she will speak about the invisible patients. A broader approach to COVID-19 is needed in data collection and research. Please, Åsa.
Okay, hopefully everyone can see what I've shared on the screen now. It's working. Okay, my name is Åsa Kristoffersson Hedlund, and I'm the chairman of Svenska Covid-föreningen, that is a patient organization for the people affected by COVID-19 in Sweden. I'm going to talk about the invisible patients. A broader approach to COVID-19 is needed in data collection and research. First of all, when we're talking about public health, we cannot only debate how to reduce the risk of people getting infected, we also need to care for the people that do get infected. So that is our objective for our association. How are we managing the needs of those infected? First of all, I would like to I think that we should notice that we have a narrative that we need to think about. Of course, this is changing everything. Everyone don't think like this, but it's important that we realize that there are people that think like this. We have preconceived notions that the disease only lasts for a couple of weeks, and we have many, many members who are sick for more than 6 months. We have a preconceived notion that if you're not hospitalized, you are okay. And that's not at all true. We have people that fear for their lives at home. Not everyone, but there are quite a few that do that. We also have a preconceived notion that COVID-19 is not dangerous or debilitating for long if you're young or previously healthy. And that is not the case either. We have members that are children and young and healthy, as well as older and people with underlying conditions. But it's also not a deadly disease necessarily if you're old or sick. That's also important to realize. And it is not a respiratory disease when we're talking about the long-term effects, especially. We're talking about multi-system disease, or perhaps even different multi-system diseases. So, and the last thing I want to say, that's very important, the doctors aren't always the experts. Of course, we respect the doctors and we know that they know a lot, but it's very important to realize that the doctors are the experts because there are research and there are combined experiences. And if we don't have that, we don't have experts. And that is something that we have to remember, we have to be humble about. And if we have humble doctors, they need to think about their learning curve. And this is what we as patients see. We need to compile data, but also we need to distinguish two groups. We have patients that are hospitalized and we have patients that are sick at home. And when we're talking about the patients that are in the hospitals, we have a very steep learning curve. We have a defined patient group and we have research and effective learning, that is multidisciplinary discussions and doctors that are specialized in COVID and that that compile experience, and that we find that very effective. But when we're talking about the patients at home who often suffer from long COVID, we have many of the patients. We have many patients that aren't properly diagnosed at all, which makes it hard to collect any data at all or experience. We have a lack of effective learning. That is because, like, a doctor may have a couple of cases with long COVID, so there is no specialization or coordination or compiled data or experience about this group, and that is a problem because then we don't have the possibility to learn. And last but not least, there is no research at this time, or hardly any research about long COVID patients in Sweden. And if we don't have research, we don't have experts, and that we need in order to help this group. But we're not only lacking data when it comes to how to assess patients and treat them, we also lack statistics that show us the reality that this is that we need to address this problem. I have compiled here just two statements, one from Anders Tegnell at the Public Health Agency of Sweden. He said on CNN about long haulers and sequelae from COVID-19, yeah, I mean, I don't think we really collect the data around that. Och Thomas Lindén of Socialstyrelsen, he said, it's impossible to say how many long haulers we have in Sweden. Among other things, among other things, because the group is not uniform but consists of a number of different groups with different problems. Of course, we agree with that, but just giving up and not trying to assess how many people we have that are affected by this is not a way forward, we think. we think that, let's see, yes, we are missing the broader picture, because we have to realize that if we don't collect data about all of the patients, we might be mistaking complication for the disease itself. The early complications might be viewed as this is COVID-19, and other complications of COVID-19 might not be viewed as the disease at all, and that is a problem. We see that many patients are being dismissed because of that. And we are also seeing that there might be some long-term complications that are being mistaken for a recovery phase. We see in our members that a lot of people are actually getting worse over time, not getting better. And this is very important to acknowledge, because these patients need help. Of course, we're not talking about everyone, but we're talking about a significant group. So if we're looking about at these three phases, falling ill and acute respiratory phase and long haulers, we see that the learning curve is steep when it comes to the first two, but when we're talking about the long haulers, patients are often being dismissed because they are incorrectly being compared to complications of the acute respiratory phase. And they have sometimes very increasing debilitating symptoms. We're talking cardiovascular, respiratory problems, fatigue, neurological symptoms, fevers, muscle weakness, and more. So how did we get here? What we can see now is that in Sweden, the narrative of long COVID is changing, but we are skipping a step. We have talked about, we've had an expectation of rapid recovery, but when we didn't see the rapid recovery in some patients, we started talking about the risk of chronic illness. But we skipped a step, that's the step of trying to stop the development of chronic symptoms. And that's what we are trying to change, because we're talking about a lot of people that should be receiving help to get better. So how do we try to stop this development? We need a broader perspective on data collection and research in order to understand the full effect of COVID-19. So all patient groups must be included. And we can see like three important steps. We need to identify the patients. And if we can't do that with tests, because we have problems with the PCR tests that are often falsely negative, and the antibody tests that aren't sensitive enough to find antibodies always, even though they might be there. We need to find other ways to diagnose the patients. And we need to gather the patient and different specialists in long COVID clinics to facilitate learning, because then the doctors can have many patients and they can be specialists. This is very important. And we need to fund research for long COVID specifically in these clinics where the patients are easily accessible. Without funding, especially for long COVID, specifically for long COVID, I mean, we will not see any learning, we think. So can we afford not to act? We only have one randomized survey in Sweden on long COVID, and that is Novus. And they estimated that 150,000 people may be affected. And that is 1.5% of the population, and we still, the virus is still spreading. So this is, of course, very important. It affects our economy, it affects our way of living. This is possibly a health crisis and something that we need to address quickly. So I'm hoping that everyone who watches this realizes that this is something that we can't ignore anymore. Thank you very much. If you want to learn more about us, then you can go to our website, covidforeningen.se. Thank you.
Thank you so much, Rosa. This was very interesting, and certainly a group that we have to think more about. Do you know what fraction of your members have confirmed antibodies?
No, actually we don't. We have a big problem, because we've only existed for a little more than a month, and the problem is that we can't collect any data at this point. So this is a big problem for us, and we are trying to collaborate with organizations in order to collect data, not just for us, but also for researchers, so that we can learn more. Because this is a problem. But there are many, I can say, that don't have antibodies. But I think sometimes it's because of the tests also, not because they don't have them.
Another question is if they are more often women, or is there a bias? Or
there are more often women, but there are not as many as you might think if you read the media, because in Sweden, the people who are sick, they found each other on Facebook. And Facebook, we have lots of women in their 40s, 30s and 40s and 50s. So you might think that there are lots of them, but there are more women than men, of course. I think, I'm not sure, but I think like 2 out of 3 are women, around about that. But we're getting new data every day about this.
And just a final comment, Aris Mustakas writes that it is important that Åsa goes to Swedish TV.
Okay, I will try. Apply for a job. No, hopefully we will have the opportunity to discuss this in many ways.
We are very glad that Staffan Normark has agreed to say a few concluding remarks. He is now the chairman of the Royal Swedish Academy of Sciences, a newly appointed COVID-19 expert group. Please, Staffan.
Okay, I hope you can hear me. I would like to thank all the speakers today for a most informative and stimulating morning. Yes, I'm been asked to lead an academy-formed COVID-19 expert group. The intention with this group is not to evaluate what went wrong and what went right in the Swedish handling of the pandemics, but instead try to build a, let's say, a knowledge platform so that can help to be better prepared for future pandemics that are sure to come. So in that vein, I have a request to all the speakers. I would very much like to get all the PowerPoint presentations sent to me, and hopefully also 1-page abstract plus a few references. Thank you very much. Yeah, in the list of Swedish disasters by death toll, you can find them on the net. The Black Death is number 1. It killed 2/3 of the Swedish population, 1350. Spanish flu influenza is number 3, killed 37,000 people or more, about 1% of the Swedish population, 1918 to 20. The majority of these people are believed to have died because of bacterial co-infections, as antibiotics were not discovered at the time. COVID-19 is already number 6 on the Swedish disaster list, with 6,000 victims 9 months into the pandemics. A number of discoveries and technological advances in between these 2 pandemics, these 100 years, they are really the fundamentals that have given us the deep knowledge on the virus itself, how to detect it, how it infects and causes disease, and also allowed modern intensive care medicine to save life, which they have done in Sweden quite a lot during the spring break. But yet we have much to learn, and we just heard about those that get a long-term disease by COVID. Other diseases will need to learn much more about these type of diseases and how to treat them and to prevent them. And we also have to develop, even though we have diagnostic tools, but we need more, better and faster diagnostic tools, robust, cheap, available out there for early mass screening at the early stage of infection, perhaps even before individuals get disease. And likewise, we should have simple procedures for environmental surveillance of the virus. And we heard about that in water. We need to have this in various indoor and outdoor settings as it becomes more clear, at least in my opinion, that the virus can spread, especially indoors in aerosols and through the air. We need more sensitive and simple ways to perform antigen tests, and also more accurate antibody tests are needed, even though they are good, but that's exactly what however, we still have no way to know when such treatment is needed. Computational tools based on machine learning and AI are needed to make the proper models and outcome predictions that respond accurately to the mitigation strategies that are put in place by the society. And I hope also research will lead to the development of antivirals attempts that we heard today that will allow rational treatment of SARS-CoV-2 infections, but that might also work as structural platforms for new pandemic coronaviruses. And my personal belief is that we in Sweden should also think about starting a modern vaccine development platform that can be applied to any forthcoming pandemic virus. So it's clear that what we need in the future is first of all, scientific and technical preparedness. And you are really part of that. And I congratulate you for all what you're doing. And the other issue is how all of this should be coordinated and plans on how it should be governed. All these are necessary ingredients if we will get any strategy in the future to combat pandemics caused by respiratory pathogens, other pathogens. We are sure that these are going to continue to plague us in the future. Thank you very much. Thank you for the day.
Thank you, Staffan, and thank you to all of the speakers. It's been a fantastic morning, I think, with so many new lessons learned. And I would also like to thank all of the participants. You have posed a lot of very interesting questions. And as someone remarked here, we think that these questions and the answers will also be interesting and valuable to all of us. So we'll try to gather answers to all of the questions that we did not have the time to pose in real life here, and then post them on our website and also on the Slack channel. And I believe we will also ask the speakers if they would be willing to share their PowerPoint slides so that we could share those as well, because I think this is really important information for all of us to follow the progress here. And of course, as many have said, the pandemic is still not over. It will continue, and SciLifeLab has now got another 50 million kronor from the Wallenberg Foundation to continue research on maybe some of the more important and successful projects, perhaps also open some new call. And now that time is not so pressed, we can do a more proper evaluation and really think carefully about what should be supported for the coming 2 years. And this has not yet been decided, neither the process or exactly how to do this. And we really welcome your input and your feedback and suggestions. And now up here, we have the email address. And I would really encourage all of you to think about this and come in with thoughts and comments and suggestions using the email address covid19calls@sciliflab.se. And thank you all very much for this morning. Thank you.